presentation is on Helicobacter pylori. H. pylori is a gram-negative microaerophilic bacterium that is spiral-shaped, as we can see on the right. The previous genome name was Campylobacter pyloridis. Then it got changed because of the sequence of the RNA were not matching. This infection, we can find it in the human gastric mucosa, which is in the stomach. And then this organism is a capnophile, which means it grows best at the temperature of 30 to 37 Celsius. There are two main routes for transmission. It is through environmental contamination or through human transmission. Environmental contamination means that infection can be in water and in food. In food, it is mainly found in milk, meat, and vegetables. And in water, it can be through external water supply, untreated sewage water, and consumption of raw vegetables. Usually, this happens when the hygiene is not in good conditions. And then human transmission can be through gastro to oral, which means the infection is found in the gastric juice of the stomach. So when the individual vomits, the microbe still survives. Another option is oral to oral, which means it is found in saliva. And the last one is fecal to oral, which is found in the human feces. .4 billion people have been infected worldwide. It is most common in Latin American countries. It has a percentage of 75 to 83%. On the other hand, it's lower in places like Japan, that has a 39.6%, and in the US, 17.1%. There is 4 steps how H. pylori is created. Step 1 is the infection develops in the acidity of stomach by which urease regulates. Step 2 is the movement of flagella motility to reach the epithelial cells. Step 3 is the attachment to the host cell. Bacteria interacts with cellular receptors that protect from displacement from the stomach. And step 4 is the toxic release causes the tissue to get damaged. Some of the virulence factors are CAGA, which is peptidoglycan, are responsible for translocation to the host cell. VACA, which is it helps with the formation of acidic vacuoles in the cytoplasm. TUPA, which is it provides high acid resistance and it increases the production of IL-8 in the gastric mucosa. And the last one is OIPA, which is the most important factor of the infection, and it is the outer membrane protein that increases inflammation. %80 of the people usually are asymptomatic, but some signs and symptoms could be abdominal pain that comes and goes, nausea, acid reflux, and vomiting. There are 2 ways to diagnosing H. pylori. The first one is the stool antigen test, which is the cheapest one. The second one is the urea breath test, which is the most suitable and non-invasive. So basically what it is, it's you exhale into a balloon-like bag, and then they will ask you to drink a small amount of urea solution. After 15 minutes of drinking it, you will be asked again to exhale into a different bag. So the way you can tell if it's positive or not, it's If the second sample is higher of carbon dioxide, then you tested positive for H. pylori. So when it comes to treating the infection, we always try to focus on healing the mucosa that usually is the one that gets damaged by the infection. The treatment we use is called triple therapy, which is a combination of 3 medications. Omeprazole decreases the acidity, and then we use a combination of 2 antibiotics such as clarithromycin and amoxicillin, and that usually has to be taken, all 3 medications, for 14 days. Some ways to prevent H. pylori is by having good hygiene and by using water and food that has been processed correctly. Also, it is believed that the probiotics are being used to prevent gastrointestinal infections. In conclusion, if H. pylori is not treated, it can develop other serious problems like chronic gastritis, gastric ulcers, and gastric cancer. Overall, H. pylori is considered foodborne and waterborne illness because it lives short-term in the food and water, but long-term in the human body.